DNA is made up of just four bases: adenine, thymine, cytosine, and guanine. How can something so simple contain all the information required for biological life? The answer to this lies in the length and sequence of the DNA molecule itself. DNA sequences can code for genes, which are then transcribed into messenger RNA and translated into proteins. Genomic sequences can also contain regulatory elements that turn genes on and off, or serve as structural elements that help sustain an organism. In diseases like cancer, important genomic sequences are altered, leading to changes in gene function or affecting when and how many genes are turned on and off. This dysregulation can have a significant impact on overall health. In 1977, Dr. Frederick Sanger developed the first method of sequencing DNA fragments, allowing the genetic code of DNA to be directly read. Referred to as Sanger sequencing, this technology was used when the Human Genome Project launched in 1990 to slowly but accurately sequence the human genome for the first time, a feat that took 13 years. After the completion of the Human Genome Project in 2003, the development of new technologies, collectively termed Next Generation Sequencing, or NGS, took off, enabling researchers to sequence short fragments of DNA much more rapidly. Next generation sequencing has significantly increased the capacity, speed, and accessibility of sequencing. NGS approaches can perform sequencing on millions of fragments of DNA from different samples at the same time. This is called ultra-high throughput, or a massively parallel approach. As a result, the cost to sequence DNA from a single sample is significantly less compared to earlier methods. These advances in technology have greatly reduced hands-on time required by scientists and have brought the time to sequence a human genome down from years to hours. Incredibly, 5 hours and 2 minutes is the fastest time to sequence one human genome, a record set in 2022. All NGS methods follow a basic experimental protocol, which includes 4 steps. 1. Preparing the sequencing sample or library, creating a representative collection of DNA or RNA isolated from cells. 2. Making many copies of the library through polymerase chain reaction amplification, PCR. 3. Detecting the sequences. And 4. Processing of raw sequence data through a bioinformatics pipeline for interpretation.